you're seeing here is the DNA. This view of it is somewhat typical to what you might see in the media of DNA, where you're looking at it at the more atomic scale of this double helix. But what I'm going to tell you about today is this other view of DNA. This is the DNA as it looks inside the nucleus of one cell. In our bodies, we have about 37 trillion cells, and with some exceptions, almost all of those cells have a copy of all of our chromosomes. If you were to line this up, all the DNA in all of our cells up, does anyone have an idea of how long that would be? It would take us to the sun and back 61 times, approximately. But this is just one nucleus, and this, if you stretch out the DNA there, will take us about 6 feet tall, so just about this much. That's a very high amount of DNA to fit in such a tiny space. And what I love about this picture is that you can see that it's not just kind of willy-nilly floating around in there. It's highly organized. So this DNA is, the stain is showing the DNA kind of amount. So the brighter the stain is, the more DNA there is in that particular spot in the cell nucleus. And so you can see that there are some very bright spots and some very dim spots. So what's going on in there? Well, you can think of it a little bit like how you might organize your closet. So there's things in your closet that you hardly ever need, so you might package those away in little boxes and stack boxes on top of boxes, and they're just not very accessible. So that would be these very dense areas of DNA here. And then there's other things that you need kind of all the time for your everyday life. And those would be kind of in these looser areas where they can be easily accessed. Now, it's a good analogy, but it's not quite right still, because it's your closet, so you would think everything you have in there you would need at some point in your life. But actually your DNA, since we come from one cell and become all of these diverse types of cells, if you were a cell, it would be as if all the things in your closet, you also have all the items that anyone in this room needs in their closets. Meaning, and also beyond that, at any point in their life. So you might have some baby socks, some, a beard trimmer. You don't actually need all that stuff, but the cell, because we are capable of making all these diverse cell types from just one set of DNA, it will have all these things packaged exquisitely inside the cell nucleus. Beyond that, beyond the closet analogy, you might notice that there's some very empty spots in there. Are these holes? No. In these areas, there's non-DNA stuff that's intermingling with and impacting the DNA. And what I'm going to tell you about today is, what is that stuff and why does it matter? Before we get into that, I'm going to tell you about this guy. This is my grandfather with different generations of my family. So this is me and my 3 sisters there. And I'm telling you about him for a couple of reasons. One reason is that he was a medical doctor, but like many of you all, he was also a basic science enthusiast, which means that he spent his days caring about people and wanting to find ways to help people live healthy lives. But then he also, in his free time, really cared about basic research. What do I mean by that? I mean science for the sake of knowledge. So just that we can perform science and learn things, not because it's important for anything, but just because it's interesting and you're curious about it. And he, like many of us, at the lab have the belief that if you focus on how things work, the nitty-gritty details of what's going on inside our bodies, that eventually that will lead to new innovations that lead to helping people. And so in my heart, I am a very basic scientist, but most of the time, basic scientists are kind of say, okay, well, we learned this thing and maybe in a decade, this could be useful for something. What I'm going to tell you about today is how I've been lucky in that by focusing on the nitty-gritty details of how things work, maybe we're a little bit closer to getting that idea to be useful for people's health. So I'm going to tell you about that today. The second reason I wanted to tell you about my grandfather is because through a series of connections, I want to share with you how the world is quite small. And so I'm going to tell you about those now. So part of his love of basic science led him to a deep friendship with this man here. This is Dr. John Lim. He was a professor of genetics at the University of Wisconsin-Eau Claire, which is where my grandfather lived too. John Lim was growing up in Korea, and he lived in Seoul, Korea, and he was a very prolific equestrian, and so he was set to go to the Olympics in 1952. But as you guys might know, something else happened, and there was the Korean War that started then. So in 1950, North Korea invaded down south through South Korea, and John Lim was captured. And he became a prisoner of war by the North Koreans, and they started training the South Koreans to be human shields in the war. And so John thought, that's not a good idea for me. If I'm going to get shot, it'll be trying to escape. So he escaped, and he lived in the mountains for about a month. And at that time, there was shown here the Incheon landing, and so this, so he was hiding out around where the eventual truce line is, which is the 38th parallel. And in the Incheon landing, the United States forces came up through there, and he was able to essentially be rescued from the mountains. And after a few years, he moved to Minnesota, started doing genetics research in plants, and that led him to a love of the DNA. And he spent his career looking at DNA of Drosophila. What he found was that there are little parts of the DNA that can jump around. And he was very excited about this because one of his heroes in the field that he kind of learned about when he was looking at plant genetics was a woman named Barbara McClintock. And so when he found his discovery in flies, he sent her a letter, and what she wrote back was one of his most prized possessions. And I got a copy of this letter from John Lim. And so this was in 1979, so several years after Barbara McClintock had made these same discoveries in corn. But it was 4 years ahead of her Nobel Prize, which was in 1984. And so I think this is a lovely thing to share with you all, because one thing she says to him is, where have people been all these years? Meaning, I've just been waiting for my science to be heard, and then finally someone's finding similar things in a different organism, validating that what I have been looking at is important. So now we've come back full circle, because now here I am at Cold Spring Harbor Lab in the building named after Barbara McClintock. And like these three amazing, intelligent people, I share enthusiasm and a love for DNA. But I also am quite curious about something next to the DNA. And it's shown here. This is about my 10-year plan on this whiteboard. And so if we zoom into one of it, here is what we are going to talk about today. These are called nuclear speckles. I give talks to scientists, so people who study DNA, and often at the end of my talk, someone comes up to me and says, wow, I didn't know what a nuclear speckle is. And so it's totally fine that none of you, except for people who've heard me before, have heard of this, because even people in my field who study the DNA have not heard of these. So let me explain. So to take a little bit of a step back, we are human. We are made out of organs. Our organs are comprised of cells, which have, as I mentioned earlier, cell nuclei, but it's not just DNA in there. There are protein and RNA-filled structures that intermingle and impact how the DNA is interpreted, and one very important type of these is called the nuclear speckle. And what I'm going to tell you about today is how these, by zooming in to these structures, we can learn things about how the DNA is understood that gives us information that informs how the cells behave, which then informs how the tissue or tumor behaves, and which then gives us information that tells us about human health and disease. Some of these steps I haven't quite figured out yet. What I'm going to tell you about is zooming way in onto those speckles, and then how it seems to be telling us things about human health and disease. So this is our cartoon version. What do they actually look like inside the cell nuclei? So these again this is now, you saw one nucleus first, and now this is a field of nuclei. You can see that as before, the DNA is dense in some areas and sparse in others, and at a subset of those sparse areas are the nuclear speckles. This is just beautiful. They take up a large volume of the cell nucleus, this precious real estate where all that DNA is packed into. And what's inside of them are things involved in RNA production. So they're full of proteins that help different steps of the RNA being made. So here's our typical view, our typical understanding, like when I learned in the textbooks, of how genes are regulated. We have the DNA. There are specific types of proteins that bind specific sequences in the DNA. And there are a lot of different types of these. So there'll be some that are specific to your brain cells or your skin cells or your intestines. And they will inform what genes are expressed and what RNAs are being transcribed. This is true, but we're missing a key component, which is this is happening at thousands of locations inside the cell nucleus. But the information we're missing here is what is the importance of where this is happening relative to these nuclear speckles? You would think that if these nuclear speckles contain all these factors that help produce the RNAs, that it might matter if this was positioned at the speckle or away from the speckle. That's what I thought too. And I thought that it should be regulated. So I thought that if we have those in there, they're probably used and probably manipulatable. So the DNA, maybe it moves back and forth depending on the needs of the cells. And I thought, well, what might do that? And what made sense to me was that it would be transcription factors. So in a series of studies, I found that it's true. Transcription factors, things that specifically bind particular parts of the genome, reposition the DNA relative to these nuclear speckles. I mentioned earlier there are hundreds of transcription factors present in the different cell types doing different things throughout our bodies. So far, we've found this activity with 3 of those hundreds, because that's what we've looked at so far. What's interesting about them is that those 3 had a little part of the proteins, part of their sequence, in common with each other. And so we call this the speckle targeting motif. So while we've only found 3 so far, we found similar speckle targeting motifs in many transcription factors. So our idea is that this is something that happens beyond the 3 transcription factors that we found and is generalizable across many different contexts. One of these transcription factors, and this is the only thing I'm going to ask you to remember more than a couple slides, one of these is called HIF2-alpha, which stands for hypoxia-inducible factor 2-alpha. So HIF2-alpha is one of these transcription factors. Overall, what idea this gave me was that this seems important. It seems that because transcription factors regulate this to change RNA production, it's probably important across many layers of biology, and it's probably important for human health and disease. And if so, I should be able to find cases where these speckles are important for human health, and moreover, beyond just the DNA changing position relative to these guys, maybe the speckles themselves can change. So if the speckles can change, then that will affect how the RNA gets produced, which will then affect how the cell behaves, how the tissue behaves, and our overall health. And so I looked into this and found, actually, in cancer, there's 2 types of states that these nuclear speckles can take on in cells. And I call them normal-like because this looks more similar to like the non-tumor tissues, or aberrant. And these look less similar to normal tissues. So speckles can have 2 major forms in human cancer, either normal-like or aberrant. And this now gives us a handle to see, is this important? Does it have consequences? And particularly, does it affect survival? So many cancer types show these 2 forms. But when we look at survival, we looked in several different cancer types. How you look at these plots is that it's the probability of survival over time. And these 2 lines are either the normal-like or the aberrant speckles. And the aberrant is orange. If they overlap, that means that there's no difference in survival of people with those tumors with the normal-like or aberrant speckles. So here, melanoma, there's overlap between those lines. It doesn't seem to make a difference. Here in breast cancer, same thing. But in renal cancer, there was a dramatic difference in survival, depending on if the person's tumor had aberrant or normal-like speckles. Why is this? What's special about renal cancer that's different than other cancers? Well, luckily, other people have done a lot of research on renal cancer, and we know that HIF2-alpha drives renal cancer in ways that it doesn't the other cancers. So what our interpretation of this is that by combining what the speckles are doing with HIF2-alpha, which is a transcription factor that changes this position, this is what's driving poor outcomes in renal cancer, but not the other cancer types. This gave me an idea, which is, before I tell you about that, I want to tell you a little bit of background about renal cancer. HIF2-alpha is driving the tumor growth, and it's affecting the tumor in multiple ways. So it's like inside the cells, it's kind of creating things that help the cells grow, And it's also influencing the vasculature of the tumor. So it's helping blood get in the tumor, that's feeding the tumor, and it also has some impacts on what immune cells are nearby. These are different targets of cancer therapy for renal cancer. And so my idea was that, well, if HIF2 alpha is combining with speckles and the speckles are changing what HIF2 alpha is doing, then maybe that will change the nature of the tumor so that it will respond differently depending on which parts of the tumor are targeted by the systemic therapy. So we were able to test this by looking at publicly available data, and what was fascinating was that if the speckles were aberrant, they showed better responses to therapies that targeted the cancer cells. On the other hand, if the speckles were normal-like, they showed better responses to treatments that targeted the immune system. What this means is that by combining our knowledge of fundamentally what speckles are doing with kind of how patients respond, we can now help potentially sort patients into groups who would respond better or worse to particular therapies. So this helps solve a major issue currently in renal cancer, and generally a lot of cancer face this problem, which is when you're faced with regional or distant metastases, who would benefit from systemic anti-cancer therapy? So this is as probably a lot of you know, if you've known or experienced going through cancer therapy, it can be quite harmful to the patient to just go through the therapy. So can we think of ways to help sort who would most benefit from getting the therapy? And if we say, yes, you should be receiving therapy, can we use speckles to decide which therapy to give? And so this is part of the future direction of where I hope to go with this research. So in total, I've told you about this human side, response to therapy, better or worse outcomes, and I told you about a deep dive into what's going on with the DNA, the speckles, and how those are interacting and interfacing with each other. And the idea, and what we haven't solved yet, is how do we connect those two? So why are people with certain therapies, with certain speckles responding better to one therapy or the other? And that will have to do with how the speckles are impacting the cells and how the cells are impacting the tumor. And so that's one of our major goals now. So with that, I'd like to thank my lab members, and I will take any questions.